Let us start. Good afternoon, everybody, dear colleagues and dear guests. Today is the 12th of February, 2021, 3 o'clock, 2 minutes past 2, Moscow time, and let me declare open a session of the Dissertation Council for the defense of thesis by Lobov, Arseniy, Andreevich for the degree of Candidate of Biological Sciences, Scientific Specialty 030304, Cell Biology, Cytology and Histology, on the theme Gamete Interaction Proteins as Factors of Reproductive Isolation on Cryptic Species of the genus Littorina, by the order of the Academic Secretary of St. Petersburg University of the 30th of November 2020, number 10868/1. Me, Ostrovsky Andrei Nikolaevich, Doctor of Biological Sciences, Professor, Department of Zoology of Invertebrates of St. Petersburg University, was appointed Chairman of this Dissertation Council. Other members of the Dissertation Council were appointed by the same order, and let me introduce them to you. Luktanov Vladimir Alexandrovich, Doctor of Biological Sciences, Professor of Department of Entomology of St. Petersburg University. Yeah, you can hear us, and all is well. Dolgech Vyacheslav Vasilovich, Doctor of Biological Sciences, Head of the Laboratory of Molecular Protection of Plants of the Russian Research Institute of Plant Protection. Vyacheslav Vasilovich, yes, all is well. Thank you. And in the remote interactive mode, we have Irina Vladimировna Guzheva, Doctor of Biological Sciences, Head of the Department of Molecular and Cell Interaction of Institute of Cytology of Russian Academy of Sciences, and I think she can hear me. Thank you, Irina Vladimirovna, and Dr. Emilia Alvarez, Doctor of Biology, University of Vigo, Spain. Dr. Alvarez, can you hear us? Please say yes.
Yes.
And the applicant is also present, Arseniy Andreevich Lobov, also his academic advisor, Candidate of Biological Sciences, Senior Lecturer of the Department of Invertebrates of St. Petersburg University, Maltseva, Arina Leonidovna. I have to announce if for any reason the connection with me is interrupted, I'd ask the next person on the list, Vladimir Alexandrovich Luktanov, to announce a technical break, and if the connection with me cannot be restored, then let him conduct the meeting further according to the procedure. Vladimir Alexandrovich, do you agree? Yes, I see. Yes, I agree. Okay. I hope other council members do not mind because of this mode today. To improve for the colleagues working in the remote mode, Dr. Alvarez, Irina Vladimировna, please switch off your microphones during this session to avoid any interruptions, and please remember to turn them on when speaking. Let me inform you that our session is being broadcast online, live at St. Petersburg University website. The speeches are being simultaneously translated from English into Russian and from Russian into English as appropriate. During the broadcast of the Dissertation Council session, an email is currently displayed, and during the session all the listeners can send their opinions and questions to the applicant online. Questions should be related to the thesis and the ongoing scientific discussion of his speech. So these questions shall be forwarded to me by our technical support department, and I shall read them during our discussion. Questions should be related to the applicant's speech and the content of his thesis. No external questions please don't ask, not relate, do not ask questions. It's also necessary before speaking it's necessary to give your full name, position, and employer. That's a must. And once again, questions that are not related shall not be presented. And according to the order of awarding academic degrees of Candidate of Sciences and Doctor of Sciences at St. Petersburg University, approved by the local regulations of the university, hereinafter referred to as the order, a session of the Decision Council shall be considered competent if at least 2/3 of the approved composition of the council present, but not less than 4 persons. Our council consists of 5 persons. All 5 are present, including 3 council members in the remote interactive mode. Audio-visual contact has been established with all council members, and thus we have the quorum. Let me set forth the following procedure of today's session of the Decision Council with total duration of approximately 2 hours. Hopefully we'll manage. We'll start with my summary report on documents submitted by the applicant and their compliance with the regulations, and answers to possible questions, approximately 5 minutes for that. Next, there will be the applicant's report outlining the key points of his study. He will have 15 minutes for that. Questions to the applicant strictly regarding his report, no more than 2 minutes for each question, and answers of the applicant should not take more than 5 minutes. After that, I will present external reviews. We received 5 external reviews, and I will, if there are any, the procedure will depend on critical remarks, if any. Then I will give the floor to all the council members, and so with state your opinion, questions and suggestions to the degree applicant, and each council member will have approximately 10 minutes. After that, the degree applicant should answer these questions and critical remarks after each speech of each council member and each external review, to give the floor to the applicant and let him answer questions. Next, we shall have open discussion. All the attendees, anyone present, physically or online, may come up with suggestions to the applicant, but strictly related to the topic of the thesis. So we'll see how this discussion will go. And to ask a question, it's necessary to register yourself, and you need to give your name, as I mentioned already. Please introduce yourself and who you are, your occupation, your position, and employment. We need to know who's speaking. And next, I will present questions we have received online, if any. So I will start with real questions, live questions, and then we'll take questions sent via the internet, and answers of the applicant shouldn't take more than 2 minutes for each question. To satisfy. Hopefully that could satisfy the everybody. So after we finish with all the reviews and all the questions, we'll give the floor to the academic advisor of the applicant. And during the open voting, we will, before we vote, we will have a discussion. Sound shall be switched off, and we'll have approximately 5 minutes to agree or to disagree if the applicant deserves or doesn't deserve the degree. After that, we shall vote. So the voting shall be open, and I will count the votes, and after each council member speaks, and the results shall be recorded in the minutes of the meeting. And then we shall make a decision on awarding or not awarding the degree. And the last item on our agenda, that will be closing remarks of the applicant. We'll have 2 minutes for that. Well, dear colleagues, do you have any questions or possibly objections to this procedure? No, we don't have any objections. Thank you. Thank you. Okay.
No?
Thank you, Irina Vladimirovna. If there are no objections, let us proceed. And first of all, please switch over your mobile phones. I'm switching mine off, so nobody interrupts us. Dear colleagues, working in the remote interactive mode, please do not switch off your phones so that we could reach you or our technical officer if something goes wrong. Thank you. Well, let me start our session. First, I have to inform you that the thesis by Lobov, Arseniy, Andreevich for the degree of Candidate of Biological Sciences, specialty 030304, cell biology, cytology, and histology, on the theme protein interaction mechanism, aspect of reproductive isolation of cryptic species, Littorina, was accepted for defense by the Order of Academic Secretaries in Petersburg University, dated the 30th of November 2020. Number 10873/1. Have to say that. Lobov, Arseniy, Andreevich wrote his thesis at St. Petersburg University under the guidance of Candidate of Biological Sciences, Senior Lecturer of the Department of Vertebrates of St. Petersburg University, Maltseva, Arina Leonidovna. The number of publications which set forth the main findings of the thesis according to the enclosed list is 6, all in peer-reviewed scientific journals from Ministry approved by Ministry of Education, Russian Federation. I beg your pardon, there are no publications in journals, but all the 6 publications I mentioned in journals indexed in scientific databases, Web of Science and Scopus. Yeah, the wording is not good. The applicant submitted to the Academic Secretary of St. Petersburg University, Gnodov Alexander Valentinovich, a full set of documents to accept for consideration he defends his thesis. All the documents comply with Article 12 of Section 3 of the Order. All the documents submitted by the applicant, according to the information I received from the curator, comply with the regulations and are stored in the applicant's attestation file. Copies are available from the officer of the Dissertation Council Activity Support Department, who is currently present. Before I give the floor to the degree applicant, there's a question. Do you, dear council members, have any general questions to the applicant, and is it necessary to review the entire list of documents submitted by the applicant? I don't think that is necessary. Ekaterina Vyacheslawna is here, is responsible for all the documents, and you can check on her if it is necessary. I have a question. May I check something? What about the Web of Science and Scopus? How many papers in one all index in Scopus and 5 in both? I see. So, Scopus 1 goes with Scopus, and 6 in Scopus, and I see. Thank you. I have no more questions. Okay. Arseniy, Andreevich, you have the floor. Please, dear Chairman, dear Council members, dear listeners, online and offline, I'd like to present to you the main results of my thesis for the degree of candidate of biological sciences. The problem of species remains one of the most discussed issues in biology. Recently, the biological concept of species has been supplemented with ideas about phylogenetic, evolutionary, ecological species, and finally about the unification of species based on their recognition. That does not remove the relevance of analyzing the key features of species as genetic unity and isolation from other species. That's why the formation of reproductive isolation remains one of the central issues of the evolution of biology. However, the mechanisms leading to efficient subdivision of gene pools and to speciation remain highly unclear. This is especially true for mechanisms that provide interspecies barriers. Analysis of this issue is impossible without the use of convenient natural models. These can be systems that are closely related, recently or sometimes not completely divergent species that form complexes of closely related populations living in symmetry. Such group includes genus Littorina, subgenus Littorina, live on North Atlantic coast, in the Barents Sea coast. This subgenus consists of two groups, Optosata group and Saxatilis group, and Littorina compressa. These mollusks are actively used as a model for microevolutionary studies. Nevertheless, the question of molecular mechanisms of reproductive isolation in this group is practically not been the subject of independent research. One of the mechanisms potentially involved in limiting gene flow between species of gamete compatibility. Fertilization process occurs similarly in all studied organisms and include a set of stages shown in this slide. Each of them is controlled by specialized pairs of molecules of the oocyte and sperm. For example, the egg attraction as receptor on the sperm surface. Interspecies differences in the structure of these molecules can lead to species specificity at various stages of interaction. With internal fertilization, gamete interaction occur in female body and processes associated with physiology. Sperm components transmit to female variety of signals necessary for fertilization, and its species specificity can also support reproductive isolation. For example, cryptids of Alonomobis, only homospecific sperm includes production of oocyte by female due to species-specific proteins. Gamete incompatibility attracts attention of researchers as one of the potential rapid emerging mechanisms. Back in 2002, an article was published where the authors summarized data on the evolution of a number of reproductive proteins. The authors demonstrated many proteins evolve at an unusually high rate. This is partly due to sexual conflict and sperm competition. The evolution of initial pairs of male and female reproductive molecules can lead to reproductive isolation in revolutionarily short periods of time. However, until now, the incompatibility has been cited only in insects. Development of new systems is an urgent task, and mollusks or Littorina can be used for this study. However, No reproductive processes have been described that are potentially involved in gamete recognition. Thus, we come to the aim of the work: identify and characterize interaction of proteins potentially involved in interspecific reproduction active acylation. The research plan is shown in this slide. It fully coincides with the objectives of the study. First of all, we confirm the possibility of heterospecific sperm contact. Next, we conducted analysis of reproductive tissues of males and identified 5 species-specific reproductive proteins. And then we tried to assess the possibility of their connection of a role in gamete incompatibility. To assess the possibility of contact between heterospecific reproductive products, we analyzed copulated pairs of mollusks in natural populations. The data obtained, as you can see, absence or presence of heterospecific populations shown in this slide. Males of all species, with exception of Littorina arcana, prefer to copulate with females of their own species. However, I beg your pardon, can't see my presentation. Yet we found heterospecific copulations with males of all species except Littorina ptussata and Littorina compressa. With maybe in case of Littorina compressa, this may be due to small sample size. Littorina ptussata appears to have effective pre-copulatory barriers, while in other species they are insufficient. The possibility of contact between heterospecific reproductive barriers and indirect data on sperm competition speak in favor of possibility of realizing gamete incompatibility, the group of species of subgenus Neretrima. To search for molecular mediators, we carried out proteomic analysis of reproductive tissues. There are 3 compartments where such proteins can be found. First, these are seminal vesicles, shown green, where mature sperm is stored. This next comes prostate, where the seminal fluid is synthesized, and penis. Penis of these mollusks has mammifold glands. Their function is still unknown, but it's probably associated with reproduction. To search for species-specific proteins in the piscine, we compare the pretomes of the mammaliform glands in 4 species. We found only 13 proteins specific to mammaliform glands, but none of which were species-specific. The slide shows a 2-dimensional lectoformogram comparing proteome of the mammaliform glands, red, and on the right you may see master gel obtained from the analysis of the 4 studied species. While mammaliform glands do not have species and tissue-specific proteins, our attention was focused on 5 proteins specific to the main fragment of the pieces, only found in Littorina saxatilis and Littorina arcana, as shown here. Unfortunately, we were unable to analyze them with spectrometric analysis. We analyzed prostate tissues in all 5 species. 9 of tissue-specific proteins were found in the prostate, 89 of which are species-specific. In this slide, you may see master gel obtained. We obtained shows the proteins in color. However, most of these 89 proteins had relatively low relative abundance, left a 4 gram below. This fragment comparing the prostate proteins, red proteins, and Littorina compressa, green proteins. The noted proteins, one of the most abundant in the prostate, and well, they have great potential for further study. However, we were unable to identify them with mass spectrometry. For analysis of seminal vesicles, instead of undirected search, we focused on the analysis of proteins, vesicular fraction of mature reproductive products. First of all, we focused on acrosomal proteins in external utilization. Our attention was drawn to 2 proteins, which most main components acrosomal extract of Littorina ptussata, and same proteins can be seen in seminal vesicles. You can see they are species specific. Yet, like other species, we failed to identify them from the database. Thus, in each of the studied tissues, we managed to find species-specific proteins which would be of interest for further study in seminal vesicles. Yet none of these proteins, so we needed additional analysis for their further study. By analogy with mollusks of Haliotis, we focused on the assumed acrosomal proteins, LOSPA and LOSPB, and on this slide you can see a diagram of our studies. Initially we were unable to identify these proteins using mass spectrometry and switched to N-thermal sequencing. As we obtained amino acid sequences, we performed race amplification and we obtained the full sequence of DNA, and we added it to the database and obtained and we were able to prove corresponds to the initial protein. Then we analyzed the structure with using hybridization, immunochemistry. We analyzed the pattern of expression and localization of this protein. In the end, we studied the variability pattern in all the studied species to try to assess the possible connection with isolation on the study. In this slide you may see a sequence we obtained with race amplification. It turned out that both proteins are homologous. The results of identification methods were similar to both proteins. Moreover, we were able to detect only one cDNA with weight of both LOSP and LOSPB. In this sequence we found two groups of repeats can be found, and between spacer regions are located between them, marked here in bold and underlined. So we suggest the two proteins with of this predecessor protein, and we will obtain two proteism with molecular mass corresponding to LOSPA and B. Search for homologs of the obtained sequence of NCD database gave no results. So apparently this protein represents a new protein family. So this protein emerged in Erythrema group is a new protein family. According to the analysis, except for and has no domain structure. Thus the function of LOSP remains unknown, but most likely has some enzymatic function, maybe a ligand or cofactor involved in signal transduction. And hybridization and immunohistochemistry stain small rounded cells in the lobes of the gonad. Contrary to our initial assumption, these cells are not related to spermogenesis as we suggested initially. This protein accumulates in large quantities and parasmatozoa large sterile cells. As you can see, these lefts are light microscopy and loose sperm, and on the right you may see the results of immunohistochemical staining, and in blue is fragmented nucleus and of dozens of euspermatozoa attached to the parasperm called blue. With detailed analysis, it's possible to detect all stages of development of paraspermatozoa described earlier at the outer structural level. This slide shows in which arrows indicate the stages of development accompanied by accumulation of LOSP in the cytoplasm. Earlier, we demonstrated vesicles apparently containing LOSP. So being LOSP is released after copulation, though the function is still unknown, and specificity can be connected with reproduction. To indirectly assess a possible involvement in reproduction between species, we analyzed the DNA sequence in all 5 species, and detailed analysis of the interspecific polymorphisms in Littorina plusata and Littorina saxatilis and Littorina arcana. On the left, there's a dendrogram obtained by phylogenetic analysis of LOSP, and on the right is schematic presentation. representatives of the two groups form two monophyletic branches. Littorina Férussac, LOSP, is conservative. At the same time, we see significant differences between Littorina Férussac and Littorina Férussac, shown in this slide. Littorina Férussac has a large 28 acids and a whole group of substitutes. And that in Saxatilis group, LOSP is highly variable and subject to positive selection. More of the species of Saxatilis group has mixed clade. So we could assume that this pattern of polymorphism is associated with the effectiveness of precopulative barriers. Males of Férussac, we never found a single copulation involved females with specific females. While in Littorina arcana, many copulate with females of Littorina saxatilis, heterospecific females. And LOSP has high variability. Yet obtained data, we don't have sufficient data to interpret the role of this polymorphism. It would be important to study the polymorphins of Littorina Férussac population, for which the possibility was demonstrated, and to carry out direct functional experiments. And on the basis of our research, the following conclusions can be drawn. So first of all, the incompatibility of gametes between species is possible, and we identified 15 proteins for gamete incompatibility, 6 of Littorina fabalis, 2 Littorina compressa, and 2 Littorina obtusata, which are 2 forms of the same protein. However, to date, only one of them has been described, LOSP. Thus, is a representative new family of parasperm protism. It begins to be increased at the early stages of development of cells and accumulates in cytoplasm, where it is localized before insemination. And between LOSP of these species, the work of Littorina arcana, and potentially this pattern can be connected with specification in this group. So we published 6 articles in peer-reviewed international journals. That's all, and thank you for your attention. Thank you very much. Arseniy Andreevich, you were on time. Now it's time for questions that both the attendees can ask questions, and anyone watching our session. So maybe we could start with the council members. Do colleagues have any questions? Yes, please. I have a few questions. My first question is, you failed to identify with mass spectrometry what is the Littorina mollusks, and maybe there are some other species the genome of which has been better described. I'm asking this question to see how this sympatry model is convenient from the angle of molecular studies. Did you get my question? Yes, thank you for this question. Let me start with this mass spectrometry issues. First of all, it's important that for all proteins I mentioned, we obtained images of high quality housekeeper proteins. We can identify them without any problem. So the problem here is only in these proteins because they are very evolutionary young. So in other mollusks, they have not been identified. Yes, I'm afraid other mollusks don't have them. As I mentioned briefly, we received transcriptome of Littorina, and they don't have any LOSP analogs. So this is a subspecies, a sister subspecies, also found in the same latitudes. The North Atlantic region, the Barents Sea coast, and in other places they exist in Cypotry. And a similar situation was found for other gamete interaction proteins. Drosophila, many nucleus potentially connected with gamete incompatibility are often absent in other insects, and lysines also found in tegular mollusks, even though authors conducted a massive study looking for homologs, but they never found them. Thank you for this exhaustive answer, this detailed answer. LOSP-A and LOSP-B, these are fragments of the same protein. How did you obtain? Did you obtain antibodies, or were these or bacterial synthesized to obtain this protein? We ordered peptide so we used a fragment of the protein, and with the help of our databases we identified the fragments we needed, and we ordered the peptide which we needed, which was converged with, and then we used for rabbit immunization. Was it LOSP-A or LOSP-B? What type of antibodies? Antibodies to sequence obtained with help of race modification. But these bodies should mark, let me show you a picture. These antibodies to this fragment, the green fragment, fragment identified for both proteins, and the underlined fragment for both, and our antibodies are fit somewhere in between. Would be interesting to obtain antibodies to these fragments, so then we could answer the question if these two fragments are present in the tissue and how they mature, but so far we have not done yet. That could be the topic of another study. And unprocessed forms we were able to identify. No, we were not. We used only blue vesicle, so probably gonads in earlier stages. Thank you very much. And maybe we should try to do this. These were all my questions. Thank you. Do we have more questions from the council members? Or maybe the attendees? Any of the guests would like to ask something? I think the council members are saving their questions till it's time to present their reviews. Ekaterina Vyacheslawna, have we received any questions online? No, there are no questions in our mailbox. I'll see if there are no questions. Okay, then let me ask, since my field is not molecular biology, and in speciation I have some experience. Could you please explain this genus as the evolution mechanism, phylogenesis, enabling production of new species? So maybe you know, or maybe you have any experience with the potential mollusks. And for example, the corals, well-known species using this mechanism actively, and the ability to copulate provides lots of possibilities. Could you please comment this It's a very general question. My question is a bit general, but thank you for this interesting question. First of all, yes, indeed, phylogenesis seems to be an important factor of speciation, and there are specialized concepts describing its role in speciation as a separate individual factor resulting from secondary contacts between populations and insect species. And it's been researched actively, the gamete incompatibility. In case of mollusks, this is a very challenging question because so far we don't know if Littorina arcana, which I mentioned, as a single group, or are these separate species? There are some research that large hybrid genesis is possible, and they can be considered from the point of speciation, and this is one of the issues of these one of problems with this model. That's why we started this work. But I have to say we didn't get any much closer to the answer. There are populations with which our colleagues, Professor Alvarez, are working, and in our populations we assume it doesn't occur, and these populations, especially in the context of this paper, how inductive proteins can contribute to species formation, speciation. So maybe in the future, when we describe a protein similar to LOSP. Thank you. Thank you. Thank you, Arsenio and Andreevich, for your answer. I got your idea. Yeah, that's indeed very interesting. And I have a person now. Professor Alvarez, do you have any questions at this point?
Well, I have one question about the thesis.
Oh, yeah.
If it's possible, I would like to know how you did the test of the synonymous, non-synonymous substitutions. It should be done comparing pairs of populations, but in some part of the text you mentioned that you use within species comparison. How does work? Could you clarify how was the methodology to use those tests?
Actually, we're not pretty sure about the using of DNDS in the case of interspecies polymorphism, and the presented data is just a bit premature, I think, and we're still looking at better ways to perform the analysis. But now we just performed DNDS analysis in the Mega software used for genetics and so on. I hope I answered your question because I'm not hearing it clearly, unfortunately.
Так, Арсений Андреевич, вы ориентируетесь? Вы поняли, что он, как нам перевели, пытался спросить?
Я не полностью услышал вопрос.
I couldn't hear the question very well, so maybe I'm not sure if I answered your question. The applicant is asking if he answered his question.
It's to me. Are you asking to me?
Yes.
Oh, well, fine. It's easy enough. I think that.
Sorry, because I heard only a part of question, so.
The applicant is saying he. Okay. Unfortunately, we can't hear the question.
I lost sometimes the.
Okay.
That's fine.
Спасибо. Да.
Okay. Okay, so the applicant can relax. Do we have any other questions? May I ask a general question? Question about the human evolution. When we all lived in Africa, our predecessors, and for a long time, we considered that Cro-Magnon humans ate up, but of late there's some data that each of us has some Neanderthal genes in each of us. What do you think? Why do genes isolated and male gonads that prevent fertilization and Neanderthal, or it occurred? So how did this happen? Because we all have these genes in us, and so we will be informed. Okay, we can get evidence of that. Thank you for this. So thank you for this philosophical, interesting question. First of all, I don't think the evolution of a separate feature coincides with evolution of species. So by gene analysis, so it's hard to describe evolution of Homo sapiens. And as far as I understand, I'm not an expert on anthropology. But as far as I know, we don't have any clear data on if we were isolated from Neanderthal or not. And if indeed this was some sort of failure and blunder. So that's why I cannot really give you a professional answer. And as for if this was a kind of a blunder, in case of gamete incompatibility, this is a separate, entirely separate topic. Indeed, gamete incompatibility often is manifested by homospecific, heterospecific sperm. Fertilization is possible, but homospecific fertilization is much more efficient when oocyte is present, surrounded by sperm. Homospecific fertilization occurs, but in some parts of population where oocyte can only interact with homospecific sperm, it's possible the more heterospecific fertilization may occur, which we show in all the models we studied. One of the classic models of gamete compatibility, external fertilization they have. So this with reproductive isolation often can split heterospecific species, but with much less efficiency. And this difference in efficiency is sufficient for species to exist separately. So a weak, small flow of genes may survive, but it's insufficient for the species to join completely. So maybe this also occurred here. Here it's important to keep in mind, I read about this, maybe my colleagues will correct me, there was much competition between between the human species, and maybe for social reasons they wouldn't copulate. So maybe this talks about, manifests some other reproductive barriers, not so much of gamete incompatibility, maybe. Are you satisfied with the answer? Yes, I'm quite satisfied, thank you. And indeed, as far as I remember, the traces of Neanderthaler traces found on the outside the center of Europe, and so that's what Arseniy Andreevich has just explained, where they don't have enough, where the population of men is not large enough. They copulated with foreign men. Does anyone else have questions? Or maybe we received any questions online? No, we don't have any questions. I see. Okay, then let's move on. And now it's my part of it's my turn now. I have to present the external reviews we received, and after that I will give the floor to the council members, and they will present their reviews of the thesis and answers to questions. And once again, let me remind the applicant it would be easier to take questions of each reviewer individually, so we don't get confused. External reviews we received from Lobov, Dmitry Sergeyevich, Doctor of Biology, Senior Researcher, Head of Laboratory of Cytology Institute of Russian Academy of Sciences. The thesis of Panova, Maria Vladimirovna, researcher of Göteborg University, Genichovich, Ignatiy Evgenovich, and Dr. Rui Faria. Let me do it as follows. Since 4 reviews of 5 have no specific comments, Zikin, Genichovich, Panova, and Faria, I will not read the reviews because they all happy with the thesis. Dmitry Sergeyevich Bogolyubov's review I will read because there are it contains many questions. So every paragraph is a review. So I suggest of the reviews is a question. So I suggest you listen to the review, and then we will give the floor to Arseniy Andreevich and let him answer the question. A review to the thesis by Lobov, Arseniy Andreevich on protein gamete interaction rows as factors of reproductive isolation, submitted for the degree of candidate of biological sciences, specialty 030304. The mixed sympatric populations of closely related mollusks, Littorina, are a perspective model to study the issue of speciation, reproductive behavior, and reproductive isolation. In modern use, a necessary stage of speciation, important criteria of identity is reproductive isolation, one of the mechanisms of which is gamete incompatibility on the protein level recognition. As the author states, gamete incompatibility can be a universal reproductive barrier for animals with gender processes. One of the most results of the work is identification with the help of bioinformatics methods of new proteins of male proteins that have no analogs and potentially can be involved in gamete incompatibility mechanisms. The main achievement of the applicant, in my opinion, is identification and early description of the new species-specific protein, LOSP. Since the author does not explain what are those isoforms or proteolysis products. Also, the author never explained failure to identify LOSP with the help of mass spectrometry. The work indicates that LOSP is located in a cytoplasmatic parasperm, special, not capable of fertilization cells. The author thinks that this protein is involved in gamete incompatibility mechanism. As invite the author to explain how, in his opinion, LOSP, being protein, parasperm protein, is but not an oosperm, takes part in fertilization and promotes the reproduction isolation. Figure 14 is stage 1 of parasperm development, shows differentiated cell. And what are the predecessor sperms of parasperm? What is their nature? And has author observed them in his material, characterizing new protein. The author says that his molecular exception of N and part is quite irregular. So what is his opinion about the functional partners? Also, the internally unorganized proteins capable of self-association and LOSP containing granules in parasperm, according to the author. What's the protein dynamic and release mechanism from these structures? And finally, the results of acid in situ hybridization shows that explains this zoning and this lobula of Y gonate. You observe the same expression. Recent research, as Domina, reference to Domina work, demonstrates. There's a list of literature that testicle of Saxatilis. What is the structure of lobule of Littorine from Optusata group, and are they similar to those of Saxatilis? Has the author observed in Optusata spermatogenic cysts and cyst cells such comparative data? Since the author analyzed 5 different Littorina species, it would be very important for better understanding of sperm genesis and mechanisms of reproductive isolation. And then the thesis by Andreevich Lobov is a complete work, has obvious relevance and practical significance, conducted at a high professional level. The conclusions are convincing, though of course some conclusions would do with some improvement. Material of the thesis have been tested and published in leading scientific journals and conferences. Some disappointment, slightly disappointed with some inaccuracies, including numerous typos. And significant figure 15 suggested by the author basis unfortunately is impossible to read. Yet these critical remarks do not affect the overall positive impression. And in conclusion, the thesis by Lobov, Arseniy, Andreevich on the theme interaction proteism as the corresponds to the main requirements set by the order, the 1st of September 2016, 6821/1, on the order of awarding academic degrees at St. Petersburg University. And the degree applicant, Lobov Andreevich, deserves being awarded degree of candidate specialty 030304, cell biology, cytology, and Article 11 of the above-mentioned order has not been violated by the applicant. Bogolyubov Dmitry Sergeyevich, Doctor of Biological Sciences, specialty 030304. And this is followed by all his titles. Well, thank you for reading the review. Also, all the 4 reviews are for externals. Well, they all have this in the same text in the end, that the thesis complies. So please answer the question. I would like to thank Dmitry Sergeyevich for such a detailed review and such close attention to my work. So I will look up my use my notes not to miss something. So the question of LOSP-A and LOSP-B, what these are, as I have said already, and this sequence with electrophoresis, nuclear electrophoresis, we identified two zones corresponding to two proteins, each of which we cut and analyzed with the help of mass spectrometry, but unfortunately we were not able to identify them in databases because a homology of this protein is emptied. With the help of N-terminal sequencing, we obtained this sequence which corresponds to from the N-terminal. The predecessor protein has cut peptide which is removed in the process of synthesis. So we obtained, with the help of race amplification, we detected just one cDNA, and then having conducted, we 5-end amplification, 3-end, backwards to 5-end, and this we obtained the complete LOSP cDNA sequence. We found only one sequence shown in this slide. Then we conducted LOSP in 201 individuals. This was data variability analysis given in the end, and in none of them we detected polymorphism, and so we always saw homogeneous product corresponding to this cDNA predecessor protein. And so we have not conducted special to explore nature of these two forms of protein, LOSP-A and LOSP-B, but they assume this is due to proteolytic maturation. So we obtain fragments correspond to, but this is a hypothesis which needs further clarification. But this is a little bit beyond the scope of my current work because the process of synthesis requires additional research. Next question is how the author explained failure to detect LOSP by mass spectrometry. Once again, we're talking not failure was not with detection but with identification. So the protein was not in the database, and we can only conduct mass spectrometry identification, and if protein is not in the database, we cannot identify it. So we obtain cDNA sequence by initial spectra. Next, Dmitry Sergeyevich suggests elaborate on the question of how LOSP, being a protein of paraspermia, still not in euspermia themselves. And this is the most difficult question because, of course, initially we hunted for in haliotis external fertilization, but in the end we found, to our surprise, we found this LOSP, first paraspermia protein described in literature earlier, had in insects, but authors never focused on modification of specific protein and remained undescribed. Sperman heteromorphism is widespread in insects, fish and shellfish. However, it's poorly understood. Especially in the beginning, many authors believed that paraspermia is disorders in the development of eusperm. As a mistake. There are some other authors who agreed that sperm heteromorphism is a natural biological phenomenon, postulates its evolutionary neutrality, or saw the cells as some cells that simply support sperm and are needed for normal sperm production, and do not exceed, mostly conducted on insects, and they on phenomenological level without any. And as for according to literature obtained by Nixon before year 2000, 3 principal functions of paraspermatozoa can be distinguished: maintenance of eusperm viability. Second function is sperm activity modulation, and thirdly, regulation of postcopulatory processes, including sperm storage. And the function associated with transport and maintenance of viability is contact with seminal fluid in the prostate. The sperm detached from prospermatogonia, and after that and the nucleus is fragmented already, and the function of the parasperm is to transfer their cargo to the female, as you can see in this slide. Thus, the function of this protein is somehow associated with the regulation of same postcopulatory processes, but it's hard to present exact mechanisms. I'll try to give you some examples from other model systems. For example, there are simulate, they do this species specific, the oocyte production, and needed for fertilization. We shown for insects and heliotis, and in litteromorphic mollusks, physitron homogenate leads to agglutination of the eusperm, and we may suppose that it may be somehow connected with, in the case of this model, Physillita molluscus, it was never got developed further, because in insects it was quite poorly demonstrated that this copulative plugs and components are able with a reproductive isolation, and in principle in whole group of such as fruit flies, crickets, and some others, many others, with head homospecific copulation, homospecific sperm is effectively replaces and sperm storage, and further will be homospecific. A similar phenomenon occurs with homospecific sperm. With heterospecific sperm, it's by far more efficient, and will be molecular with artificial symmetry and development of gamete incompatibility. Is the same genetic locuses participate as in case of sperm competition inside the species, but the authors never provided any specific markers and never made any specific molecular cascades they provided, so all these is still a bit vague. Next question is figure 14. Shows development of paraspermia and differentiated. So what about the progenitor cells? And what is their role? Unfortunately, our methods do not allow us to identify parasperm progenitor cells, and the analysis, unfortunately, we were not able to identify these progenitor cells. And the answer would be if there are progenitors, he assumed but we were not able to verify this in our study, with our study. Obviously, judging by our data, zones of U and paraspermogenesis, we don't see any, maybe separate progenitors are present, but there's a need for further study here, which we have not conducted. The next, what are about the functional patterns that LOSP might interact with? With the help of ANCHOR2 algorithm, we performed analysis to predict the context-dependent, internally disorganized protein fragments. So fragments where transition from disorganized structure occurs, interaction with some globular protein, and on the basis of this algorithm, we obtained information that the entire fragment could be such an unorganized structure. But with the help of other methods, we were not able to identify and predict such a partner for protein, maybe because LOSP is not in the databases, and all work for such prediction is based. So we, in order to identify such LOSP partner proteins, it's necessary to conduct. So we are working on this, and in the future we plan to conduct a search for proteins with which it can interact. The next question: do LOSP containing granules have membranes? As the author, so what's protein dynamic and mechanics of release of these structures? Littorina parasperms are well studied at the ultrastructural level, and we may say that these they actually have membrane. And moreover, the LOSP sequence includes a signal peptide. So yes, we believe that these granules indeed have a membrane as vesicle. They are vesicles. As for dynamics of this protein and the mechanisms of its release, unfortunately we have not explored that, and they are outside the scope of our work. Next question. This recent study, Domin and co-authors. Yes, I know the first work. As Littorina saxatilis belong to a special lobular cystic type. So what's the structure of lobules of Littorine from the Optosata group? Are they similar in features to those of saxatilis? And did the author observe spermatogenic cysts and cystic cells? And so we actively follow the work of Sergei Yurievich Domin, and the second work because we made no reference to the second work because it was published after I submitted my thesis. But comparing this his data with our data, because Sergei Yurievich is using different methodology, as spermatogenic cysts, not well visible, yet figure on slide 16 shows that groups of para and ura spermatozoa at the easily visible can be interpreted by nuclei. They are located in compact groups, which can be interpreted as spermatogenic cysts. But our methods give us no opportunity to see them. And finally, speaking of typos and mistakes in the thesis and low quality of figure 15, so unfortunately, the font is a little bit small. So thank you. So I will try to keep this in mind in the future. Hopefully, I answered all your questions. Thank you. I hope that the attendees enjoyed the answers. I think we satisfied our colleague highly respected, and some questions are very interesting, and the answers to questions demonstrate the level of skill, and so you see how well the applicant has mastered his field. Ekaterina Vyacheslawna, maybe one last time. Maybe we received any questions online or via the internet? No, we have not. So then I will, since we are starting the reviews, let me invite Arsenio Andreevich to sit down. So you must be tired. And now the Dissertation Council members will present their reviews, and we'll start with Dr. Luktanov and Dr. Alvarez. Many comments are similar because we are all reviewing the same text. So it's up to the council member. You may want to read the entire review or only the critical remarks, most essential critical remarks. And also, please read the final paragraph, because we need this paragraph on compliance with all the regulations. And Vladimir Alexandrovich, thank you for the opportunity to present my review. And since I am the first council member to speak, my review will be quite complete, except I will read the formal parts as well. And I will start by saying that the thesis by Arsenio Andreevich is aimed at relevant issues of modern biology, which is poorly studied, interaction of gamete interaction that can ensure reproductive isolation and thus be factors of speciation. Let me start with more formal. The work includes introduction, 5 chapters, conclusion, references, and 3 short annexes. And its main points are presented in 6 articles. I never saw such a list of excellent journals where the articles were published. And the field of this, the sphere of this magazine, specialization of this mechanism, is as experimental zoology and ecology. And the thesis is 138 pages long. First 70 pages, that's Russian text, and well, the rest is the English version of the thesis. In the introduction is very traditional, explains relevance, aim and tasks, novelty, theoretical and practical significance of the thesis, the main provisions set for defense. The first chapter includes literature overview, consists of 2 parts. First part includes analysis of general speciation theory and gamete incompatibility as a factor of prezygotic isolation, issues to which the thesis is dedicated. The author also describes the modal system under study. Chapters from 2nd through 5 contain the author's results. They are quite well written, except for one illustration, and they provide a good impression of the author's work as results and conclusions. Also described in the articles published by the applicant. Methods and materials section explains, demonstrates that material sample is representative, and the methods proteomic analysis, genomic, immunochemistry, molecular phylogenetics, bioinformatic analysis are diverse, up to date. The author obtained an impressive array of new data. Particularly impressive is the results for gamete specification capacity. And here I'd like to mention the personal contribution of the author to all these complex studies conducted, the help of a wide array of methods, including field work, And this combination of high-quality field work enable the frequency of non-specific and species-specific copulations and make conclusions on the reproductive isolation. And at present, when the number of purely molecular works is increasing, such a wide approach we should certainly welcome. Also, what's important, the author developed a new model system that could become the basis for in-depth study of post-copulatory mechanisms of reproductive isolation. This type of isolation is little studied. How close species can coexist in symmetry. So the significance of this model cannot be overestimated, but as any good work, the applicant's work leaves us with many questions, but such a model system can enable us to move effectively. And speaking of critical remarks, I'd like to say that the species issues described from the position of typological and biological concept of species, and where the biological concept wins over. But in reality, today the discussion is mainly between the biological and phylogenetic concept of species, and the actual species delimitation is mostly occurs from the angle of phylogenetic concept, as such articles as Dicaris and Mallet, which provide contemporary ideas of species, far from species as unity of gene pool. Here, I'd like to say that the applicant has partly answered this question earlier, and symmetry and mechanisms for pre- and post-zygotic isolation do not mean the symmetry can and probably is secondary, but this idea is not very clearly stated in the thesis. And so maybe, so here we're dealing with sympatric. Maybe this still needs further evidence. And in the theoretical section of the thesis, the author never mentions species as that can identify conspecific units and recognition of species concept. But this concept exactly describes this type of species system. The synthetic theory of evolution. So this concept characterizes by Charles Darwin and experimental sciences genetics, which occurred in 30s and 50s of the 20th century, and in broad sense, the term synthetic evolution theory makes no sense as any science at the modern state is synthetic, because it's based on constant synthesis of all the new and modern ideas. And geographic isolation usually there's no contact, so there's nothing to discuss. Also, I'd like to say that all my critical remarks they apply to the methodology and cannot be applied to the main conclusions obtained by the author, and I am absolutely convinced that the conclusions are valid. And on the basis of this, I believe that the thesis by Lobov, Arsenio, and Andreevich on the theme Gamete interaction proteins as factors of reproduction of cryptic species of Littorina Férussac corresponds to the requirements set by the order 6821/1 on the order of awarding academic degrees at St. Petersburg University, and the degree applicant Arseniy and Andreevich Lobov deserves being awarded the degree of Candidate of Sciences, specialty 0304 cellular biology, cytology and histology. Article 11 of the above mentioned has not been violated. This is my opinion. Thank you for your attention. Thank you very much, Vladimir Alexandrovich, for such a detailed review. Arseniy Andreevich, I would like you to comment at least the most essential remarks made by the council member, though many things have already been discussed. Thank you for your review. I'd like to start that our work started as evolution, and our results are quite far from to argue with modern evolution ideas of evolution, because we are just getting there now. So we obtained interesting protein, but this data is still insufficient. So obviously this data is primary, and so we need to use more appropriate models, maybe study the type of Littorina Férussac, study it in other populations, and compare with our populations, and etc. And that is why in the literature overview that's why it's maybe given in simplified form. As for the question, symmetry can be secondary. That's a very interesting issue. In case with gamete incompatibility is important, but the species that currently living in symmetry are not. And this factor is a key factor, and maybe is a series of works of Ziegler, 2004, where he sums up his own research. So they have studied over 6 different species, 6 different species of echinoderms, but at the same time 3 species of echinoderms. There's no reproductive barriers, and yet secondary symmetry is also important, and there are some works on Drosophila species, where the role of gamete incompatibility is pre-coupled isolation, and gamete incompatibility occurs later, and perhaps from this point the evolution history can play a major role, and so it would be discussed. So yes, that would be interesting to discuss, and of course I'll take it into consideration in the future. But in our work, this aspect so far has no relevance for our results. Also, I'd like to thank you for taking this discussion in the field of evolution. I don't quite agree with it's a theoretical. First of all, synthesis term is mostly used, and in most modern works we use it quite often. And here I would like maybe to mention 2 works. Work of Kevin Lund, Evolution of Synthesis, and work of Golod. In both works, the subject of should be Laland and here you may see the general scheme of the evolutionary biology as seen by him, and so the elements to be added are marked with red here. So the talking about the evolution theory, because the concept is still widely used by many authors. So I don't want to discuss, wouldn't get into the discussion how correct. So one thing I would like to say is that some authors are still using the evolution theory nucleus. So maybe it would be premature to take it to a history of science. Well, I think I have answered. I believe I have answered all your questions. And in conclusion, thank you for this geographical isolation. Where it shouldn't be. This, you're absolutely right. This was a careless mistake. Thank you very much. Vladimir Alexandrovich, are you satisfied? Yes, in general I'm satisfied. I think that any good science is synthetic, and we should rely on our predecessors. And obviously we don't have to say that our work is synthetic. There's just one point I'd like to make. As any good modern work, it's synthetic. But I'm satisfied with your answers. Thank you. If we have, we can discuss these general issues later. During the open discussion, but now let us continue with the reviews, and I would like to give the floor to Professor Emilia Alvarez. Professor Alvarez,
hello.
The floor is yours.
My name is Emilio Roland Alvarez. I am professor in the University of Vigo. I will give a summary of my assessment instead of read everything, and perhaps, of course, the final assessment as well. In general, I would like to say that the thesis is particularly interesting because it focuses on both pre- and post-copulatory reproductive isolation mechanisms in several species of the genus Littorina. In particular, the detection of one candidate reproductive isolation protein, loss, of putative postcopulatory effect is of special relevance. The thesis uses most methodologies used to investigate this kind of mechanism, from DNA to proteins, and includes a study of estimation of selective effects at the DNA level by using the non-synonymous to synonymous ratio of SNPs polymorphism. Perhaps the only partial criticism is that the thesis has tried to investigate too many topics at the same time, and so in certain parts they have not enough time or resource to check deeply all or part of the alternatives. Nevertheless, the study in general is of high interest, and their publications are expected to produce a relative high impact in the field. I should comment below the different chapters, but I will not do it, and go to the end, to the final assessment, that say this thesis and the published work on the authors that summarize represents a substantial body of novel and informative work carried out for several years, either alone or in collaboration with colleagues, thought a combination of fieldwork, capture of copulates between species in situ, studying the divergence in RNA and proteins for several tissues, and identifying its functional relevance indirectly by measuring the evolution ratio, non-synonymous to synonymous substitutions. They made a significant contribution for understanding the evolutionary mechanisms contributing to species identity via reproductive isolation, in particular focusing on postcopulatory mechanisms, which has been rarely studied in the past, a contribution that would be considered to meet the criteria for the degree of doctor in biological science in any institution. This is all from my side.
Thank you very much.
Thank you very much. Thank you, Professor Alvarez. Andreevich, do you want to answer, maybe take your revenge? I would like to thank Professor for taking your time and reading my thesis, giving me some, and for your comments. I agree that some parts of my work are not maybe premature in case of analysis of observed privation, and we will continue this research in the future, and I hope that we will find more appropriate methods for analysis and to find more convenient ways to discuss our subjects. Thank you. Thank you. Hopefully, your comment, you heard Arseniy's comment. Next, I would like to give the floor to Dr. Dolgich and Arseniy Andreevich. Please have a seat. You must be tired already. Vyacheslav Vasilyevich. Well, I will keep it short because the other reviews already shown us the general picture. We see here we're dealing with a very high quality work, thesis with many international publications. The relevance of Arsenio Lobov's work is manifested in the fact that on the one hand we are dealing with the issue of reproductive isolation, especially in sympatric populations, and on the other hand the author suggests a very convenient model for this study of this issue. In recent years, a number of data on molecular biology has taken this discussion of speciation and reproduction barriers is taken to a new level. So this model is very interesting because it's talking about several species, about sympatry, and judging by his data, this model indeed is very interesting. The thesis consists of 5 chapters. The first chapter provides literature overview. The second chapter is dedicated to pre-copulative barriers, which in this model were not efficient enough. And the author manages to demonstrate that there are post-copulative barriers and mechanisms of those post-copulative barriers in this model and in most other species of invertebrates have not been studied before. One of the important mechanisms of this post-copulative isolation is based on gamete interaction proteins, and the interaction of these proteins results in phenomena of gamete incompatibility, which defines these barriers. In chapter 3 of the thesis, dedicated to this post-copulative isolation connected with gamete incompatibility, the author identifies species-specific proteins and conducted proteomic analysis of 3 compartments: vesicles, prostate, and penis. As a result, the author managed to identify a number of tissue-specific proteins, and 15 of these proteins are species-specific. And though there is no data on identification of this protein, mass spectrometry. In chapter 4, the author describes the study of the most interesting protein, which he calls LOSP, and he has demonstrated quite well that this protein is connected with fertilization. First of all, the author managed, with the help of sequencing, to determine the end and identify and decipher its full-size copy. And in addition with that, the analysis of amino acid sequences is an N-sense signal peptide responsible or transport to some compartment within the full-size replica. 5 repetitions and of such unorganized structure, the end of alpha, and obtained data that this full protein can mature into 2 forms, LOSPA and LOSPB. And a major part of the work is dedicated to the study of distribution of nuclear protein distribution in mollusk tissues and Gamete, and while the LOSP coloring identified in mature parasperm in vesicles and parasperm Gamete progenitors, and LOSP contained in the last chapter dedicated to analysis of internal and external polymorphism, and the author in Littorina Férussac group and Arcana, where precopulative barriers were least efficient, this high level of polymorphisms observed. At the same time, of Littorina Férussac, the highest efficiency precopulative is coincides with the lowest level of internal polymorphism. Such polymorphism pattern is similar to other gamete incompatibility proteins, which indirectly points at LOSP with gamete incompatibility. Yet the author himself emphasizes that this data is insufficient, and this issue requires further study. So the work is very novel. It leaves us with more answers than questions, and so this is only the first stage of working with this very interesting system that will enable the author to get answers to questions to which he doesn't have answers. The study is multidisciplinary, and this is also demonstrated by journals where the results were published. And among the drawbacks, I think the typos already mentioned. The typos were already mentioned, and I have, from the molecular point, this is not a critical remark, but a suggestion, because the author is just starting his work, and he will certainly take this into account in the future. He thinks that these 2 proteins, LOSP-A and B, they are the final functional form, and in my opinion, it would be best to study the structural features, keeping in mind that in the mature state functional molecules, let's not talk about LOSP protein is one entity. So we're looking at 2 low molecular proteins, 2 small proteins. So I will not read I won't say that the thesis corresponds to all the main requirements set that exist for candidate's thesis. And let me say once again that, in my opinion, this is an excellent work, and the author should certainly be awarded the desired degree of candidate of biological sciences, specialty cell biology, cytology, and histology. Thank you. Thank you, Vyacheslav Vasilyevich, for your positive, and I hope Arseniy Andreevich will answer you. And I would like to ask one, have you sequenced the second fold by Edmund and the ends are identical? Well, that's very interesting. Thank you, Vyacheslav Vasilyevich, for your review and for your questions. Indeed, your questions are very important, and so what we can do in the future is go study the prostate proteins, which we found. And first of all, we have sequenced and to increase the efficiency and secondly would be we will continue better, we will improve the analysis of its variability because our analysis was too broad. We identified certain phenomena we would like to understand better, and of course the structural work with protein methodologically for us is maybe the most important, the most challenging part, because we have not come across. So thank you. I fully agree that we should study these two protein forms. It's interesting that Karyotis mollusks, lysines, they also exist in two forms, but in their case these are two separate genes resulting from replication, which are slightly different, and only lysine 16 kilodaton is in oocyte membrane connected with other processes. So this is another reason why until so they thought this. So hopefully we can study this in more depth. Thank you. Thank you. Have a seat, please, if you like. Let us proceed to give the floor to Yelena Vladimirovna Guzhova. Irina Vladimirovna, please. Thank you. I will not. I'll try to make it short. And in general, I enjoy this thesis. I like the logic. It's very logical. It's well written. Like this story with antibodies, how the peptide, you get peptide description and link to and obtain antibodies. This peptide, this is that's very good, and I don't have any critical remarks here. It's very logical. It's a very mature paper with the philosophical component, which many works are lacking today, unfortunately. But one critical is I have only one remark regarding the structure of the thesis. All looks well, but methods and materials chapter follows acknowledgements. Maybe it is required by the university, but for this order. But even if there is such a requirement, you should not put the most important part of your thesis, and the results should be presented in view of this requirement, because many details of what you did and how you did, the description of methods. So this is given under methods and materials. So you wonder why green and why you use red and green. But this is maybe my only critical remark, and I don't have any other critical remark. The work is interesting, has novelty, performed on a high level, and as a result, let me tell you that the thesis by Lobov, Arsenio, and Andreevich on the theme Gamete interaction and proteins as factors of reproductive isolation of cryptic species Littorina Férussac 1822 corresponds to the main requirements set on the 1st of September 2016 order number 6821/1 on awarding academic degrees at St. Petersburg University. And the degree applicant, Lobov, Arsenio, and Andreevich, deserves awarding academic degree of candidate of biological sciences, specialty 030304, cell biology, cytology, and histology. Article 11 has not been violated. Thank you very much. Thank you, Irina Vladimirovna, for your positive review. Let's give the floor to Irina Vladimirovna. Thank you for your review. And it was our decision to put methods and material in the end of the chapter, and well, we decided to do this not because we think it's not important. We understand this is a very important part of our work, which shows how the author can trust or should not trust the results. So we decided to do that because we understood that our work is very multidisciplinary, and there are many methods, and maybe not every reader of a wide circle of experts should be immersed, should learn about all these details. Because in full form, this part was 23 pages long. And so that's why we decided to present them in an abbreviated form, following the style of many contemporary journals that do it put this part thinking that only a small number of readers will be interested in reading this part. So maybe, hopefully, the experts who will become interested will read about our methods in our articles, where our methods and materials are described in much more detail. And let me emphasize again that maybe that was not the best decision, but my reasoning here was that it makes the work a little bit easier to read. Thank you, Arseniy Andreevich. Irina Vladimirovna, are you satisfied? Yes, I'm quite satisfied, thank you, with the answer. Thank you. And I have another doctoral student in Vienna, and the style of in European universities is now more or less unified, and well, there should be an introduction and list of papers, and then each paper names methods and materials, lists methods and materials, and so this is a generalized overview and nothing else. So that's why, indeed, in some cases, the methodology, the part on chapter on methodology, is can be presented as an. So in that situation, would be useful to put it in the beginning, keep it in the beginning. But thank you. And now I have to present my review, which I wouldn't say word by word, but we all followed the same paper, so, and we same number. The number of chapters is the same, and so I don't want to bother you with repetition. So I want to talk about something else. the thesis was my student many years ago on invertebrate zoology. This was like 10 years ago. He was my student in his first year, and his progress it's very rewarding for me as a teacher, as a professor, to see his progress and how he has mastered his subject, and how interested and how motivated he is, so now I think he's much more versed. And since we all have to read this final paragraph of the review, of course there are some typos, maybe bad language, but generally the thesis by Lobov, Arseniy, Andreevich, and the theme gamete interaction proteins as reproductive selection factors of cryptic species of Littorina Férussac corresponds to the main requirements set on the 1st of September 2016, number 6821/1, on awarding academic degrees at St. Petersburg University. And the applicant, Lobov, Arseniy, Andreevich, deserves awarding the degree of candidate of biological sciences, specialty 030304, cell biology, cytology, and histology. Article 11 of the above-mentioned order has not been violated. So, we are done with this part of our. So we have not received any questions online. So hopefully, let me ask again if all the council members are satisfied with the applicant's answers. Hopefully everyone is satisfied, everybody's okay. Next. Is any of the guests is not a member of the Dissertation Council? Colleagues and guests, is anyone present here willing to speak? You may have a couple of minutes to give your opinion of the thesis and the presentation. Do we have any? Is anyone present here willing to speak? So I can only see a part of the hall. I think no one is willing to speak. Okay then. The answers and comments by Arsenio Andreevich were very detailed and highly informative. Next, once again, I have to ask if we received any questions or reviews online. No, we have not received any. Well, that's thank God. That's very good. Excellent. Now, according to the procedure, we have the. Now it's time to give the floor to the academic advisor, Arsenio. Please tell us about the degree applicant. Thank you, Chairman. Thank you, dear council members. Thank you for watching, following us online. Thank you for finding time, giving your time in this hard time to devote attention to our work. And I will try not to bother you for too long, And let me just say very briefly that Arseniy Andreevich, I've known him for almost 10 years, as Andrei Nikolaevich said. He started working with our group in 2013 when he was in his first year. And when you know someone very well, I had the opportunity to come to know someone in 10 years. And so today I was thinking, what's the most important feature of Arseniy as a researcher? And I think that he's very curious. It's his curiosity. He's curious about anything. Whatever happens in our group, in our resource center, I worked there, and he took over my job. He has mastered a wide range of methods, and as all the reviewers have noted, as all the experts noted in their reviews, and over this period, while Arseniy worked with our group, he's a very versatile personality. He was involved in everything as a researcher, very enthusiastic, ambitious. He took part in various competitions, student competitions, was very successful. He won many awards in national competitions as well. He took part in conferences, made reports. I don't have a feeling that anything special is happening now, because I'm so used to Arseniy speaking or presenting our work somewhere. Arseniy got involved in teaching. He already became the academic advisor of a master's thesis, and he's an excellent mature researcher. He took part in field work. He has a wide range of competencies as a scientist, as a theoretician. He can write applications, make reports, so he can do anything. He can do it perfectly well, and he has all the skills required of a modern researcher. So here I would finish my short speech that he's a real star, already one successful lab already invited Arseniy, and which makes me very happy, so thank you very much. Thank you, Irina Leonidovna. Many thanks to you for such high quality product. Let me put it that way. And well, now I don't have to thank the advisor. I can do it in the end. Now, formally, since our session is, some of the council members are working in remote access mode, I have to ask if council members or other attendees have any, or maybe the applicant himself, any unanswered questions, unclear issues that we should take now. Connected, maybe something connected with working in the remote interactive mode. Please say yes or no. There's one I forgot. Prostate gland. Have anyone come across such an expression. So please stop using it. Prostate gland is wrong. There is bad language. Before we vote, we have the opportunity to take a technical break for 5 minutes before we vote, and it's up to the council members. So do we need such a break? Dear Irina Vladimirovna, do we need such a break? Vyacheslav Vasilovich, if we don't need, I think we can do without the break.
I think so.
So, but you gentlemen, do we need any private discussion? Yeah, let's continue without. We don't need a break. Let's continue. Thank you. Next. If we don't need any private discussion, then we have to proceed to voting, and at that I will ask each council member individually and openly. And now, since we're not taking a break, this is the most critical part of our session, and let me once again check that we can all see and hear each other. Can we all see and hear each other? Yes, we can see and hear each other very well. Thank
yes.
you. Now I put the question of awarding to Lobov, Arseniy Andreevich, the degree of candidate of biological sciences, specialty 030304, cell biology, cytology and histology, to open individual vote. Let me remind you that a decision of the Dissertation Council on awarding, not awarding, and a degree shall be considered positive if at least a half, but not less than 3 members of the Council present at the meeting voted for it, according to Article 23 of the Order. Well, Council Member Luktanov Vladimir Alexandrovich, your opinion. As I have already said, I see no reasons to change my opinion. I think that Arseniy Andreevich Lobov deserves being awarded the degree Candidate of Biological Sciences 0304, Cell Biology, Cytology, Histology. This is beyond any doubt. Thank you. Thank you, Vladimir Alexandrovich. Vyacheslav Vasilovich Dolgich, Council Member, your opinion, please. Without any doubt, the applicant deserves awarding the degree. And once again, I'd like to say I was very impressed by the work itself. And when I heard the applicant's answers, my certainty grew even stronger. Thank you for your attention. Thank you. Irina Vladimirovna Guzhova, Council Member, your opinion? Let me just say yes, the applicant deserves the degree, the candidate of biological sciences, specialty 030304, etc. Thank you. Thank you very much. Council Member Emilia Roland Alvarez, your opinion, please.
Yes, it's the same. I agree that he deserves the degree.
Thank you. That's very well. And me, the chairman, Andrei Nikolaevich Ostrovsky, and my opinion coincides with other council members, and I believe that Arseniy Andreevich Lobov deserves being awarded the degree of candidate of biological sciences, specialty 030304. And let me thank you for voting. Now, since according to the procedure, I have to count the votes. I see all 5 council members voted for, and there's no one has any doubt that the applicant deserves the degree. Thus, let me inform you that out of 5 council members who voted, 5 council members voted for, no one voted against, and no one abstained. Thus, the decision to award to Lobov Arseniy Andreevich the degree of Candidate of Biological Sciences, Specialty 030304, Cell Biology, Cytology and Histology, has been made. Dear colleagues, since our session, some participants of our session are working in the remote interactive mode, I have to say that do council members or anyone present anyone present online, or the degree applicant, or any of the attendees, do you have any questions regarding the procedure of today's session? I need all the council members to say yes or no. No, I don't have any claims. No claims, no comments. No claims. All is well. And Dr. Alvarez, please say if you are okay with the procedure of today's session. Do you have any comments regarding the procedure? Have any comments regarding the procedure? Please say yes or no.
To me?
Yes, please.
Sorry. Yes, no, I have no comment. I agree with the procedure. Sorry. Thus, we have only 2 items. One is closing remarks of the applicant. Arseniy Andreevich, the floor is yours. Let me thank the council members and external reviewers who found the time to read my work and discuss and took part in this session. Many of the comments I heard are very informative. I find them very informative, and many of these comments will be used in my future research, future work. Also, I would like to work the translators, and the Dissertation Council activities support officers for their work. I'd like to thank the Department of Zoology of Invertebrates for supporting me at various stages of my work, which started 10 years ago at the Department of Zoology of Invertebrates and Resource Center. They provided me with lots of help and support. Also, I'd like to thank Russian Fund for Fundamental Research, and works in this supported with 3 grants. My work was supported 3 grants from this fund, and I would like to thank my academic advisor, Maltseva Arina Lenina, and Granovich Andrei Igorevich, for supporting me and guiding me during these 10 years, finding the time to master the methods, the skills of writers' skills, presenting papers, writing papers, which I mastered thanks to my academic advisors and our Littorina knowledge group, with whom I conducted field work and with whom we worked together, and my friends and family, without whom this work would be very hard.
and who helped me at various stages for my work, and thank you for your attention. Thank you, Arseniy Andreevich. Now we may let's greet the applicant on being awarded the degree. Thank you. Thank you, dear colleagues. I thank you for your work. It was a pleasure to work with you. At that, I declare the session closed. Please stop broadcasting. Thank you.
Goodbye.